Welcome to accommodate this. For decades, scientists have hypothesized about the cause or causes for autism, and research suggests genetics account for roughly 80 to 90% of the factors leading to the development of autism. However, the remaining 10 to 20% is attributed to environmental factors, and as early as 1982, scientists have hypothesized that inflammation in the womb is one of the most significant environmental factors. If you've been on social media over the last few weeks, you may have come across posts similar to these, which announced that Japanese researchers have identified an inflammatory mechanism that is being touted as the cause of autism. But these announcements are very high level and don't provide much of an explanation of their findings. So, what exactly did they find? And did they really identify the cause of autism? These questions and more are what I want to tackle today. So now that you know why we're here, let's get into the topic for this video. Findings from Fukui U. We already know that environmental factors account for 10 to 20% of what leads to the development of autism. But when we say environmental factors, we aren't talking about the weather or being outside in nature. We're talking about things that happen inside the womb during pregnancy. But even after mothers do all that they can for their unborn child, about 3% of children are born autistic because the largest percentage of contributing factors are genetic. Regardless, scientists hope that the ability to identify autism in the womb will provide more opportunities for early interventions. In my video on autism therapies, I talked about some of these interventions, which are intended to give the autistic child the best start in life. Around 1982, scientists at the NIH suggested inflammation may play a role in autism development. Then in 2001, researchers discovered an increase in inflammatory cytokines in autistic children. And in 2005, another study found ongoing inflammation occurs throughout an autistic person's life. But the discovery of what causes an increase in inflammation in autistics has been elusive. For example, quinolinic acid can inflame neurons or brain cells, and neopterin is an indicator that cell immunization has been triggered, which usually involves some sort of inflammation. However, these naturally forming chemicals have been found to be decreased in autistic children when scientists examined their cerebrospinal fluid. At most, this would suggest a dysregulated inflammatory response, but not definitively point to an increase in inflammation. And despite this and other studies, scientists know that autistic brains contain higher volumes of brain cells during development, which takes up more space and results in the development of more connections. I talked about this in my video on brain development. However, the mechanisms behind autistic inflammation have remained a mystery to scientists. That is, until now. Scientists from the University of Fukui in Japan have made some recent advancements toward validating the inflammation hypothesis. Before I get into the details, I must remind everyone that I'm not a doctor or a biologist, but I am going to do my best to explain their findings to you, and hopefully the information I share makes sense. However, I have linked to the study in the description. You'll find that below in case you want to check out the study for yourself. Now, researchers at Fukui U started with 1,258 children who were under 4 weeks old, born from 1,138 different mothers, who had blood taken directly from the clamped umbilical cord immediately after delivery. Then at age 6, those same children were given the ADOS-2, which is the gold standard autism test for all ages, and the VABS-2. It's just another reliable autism test for all ages. Participants who did not complete the tests were eliminated from the study. Then researchers eliminated those whose blood samples had hemolyzed, which means the blood cells in those samples had ruptured. And this apparently is very common. It occurs roughly 40 to 70% of the time, but they know that hemolyzed blood samples have been known to interfere with lab test results, so they're not very reliable. These exclusions brought those number of participants down to 588. Then, in an effort to ensure that their sample size was appropriate and that cases were picked at random, also while attempting to include more children than the autistic prevalence indicated, 1 in 44 at the time that the study began, researchers randomly selected 70% of participants to arrive at a final subset of 200 participating children. That was about 106 boys and 94 girls. This is well within the standard statistical range for appropriate research sample sizes. So let's talk about a few concepts and terms that you'll need to understand in order for the findings to make sense. The first term you need to know is cytokine. I mentioned this earlier. Now, cyto means cell, and kine means movement or motion. But the entire word does not mean movement of the cell. The movement here is talking about movement of signals between cells. So the term cytokine refers to proteins that make it possible to move signals between cells. Okay? So cytokines are signaling proteins. Now, let's talk about proteins. There are about 20,000 to 100,000 different proteins that are responsible for different functions in our bodies. And it's possible that there could be even more. But what they all have in common is that they are basically molecules. Now, remember in elementary science, they taught us that molecules are groups of atoms. That's what we're talking about here too. Proteins are simply molecules with a particular structure or shape that allows it to perform specific functions. Each protein is a collection of atoms that are connected using chemical bonds. Despite there being so many possible proteins, they are constructed using only 20 amino acids. Amino acids are considered the basic building blocks of proteins, and we can break down proteins into amino acids and further break down amino acids into smaller molecules known as either carboxyl groups, amino groups, or side chains. Now, carboxyl groups contain carbon atoms that are double bonded to an oxygen atom and hydrogen atom. Carboxyl groups have a negative electron charge, which makes them mildly acidic and susceptible to donating or giving away hydrogen ions in order to form bonds. Amino groups contain nitrogen atom bonded to two hydrogen atoms. And amino groups have a positive proton charge, which makes them susceptible to accepting or attracting hydrogen ions to form bonds. Then there are variable groups of atoms known as side chains. These are often represented with an R, so scientists sometimes refer to these as R groups. These have no single configuration. However, when a carboxyl group connects to an amino group and an R group, it forms a full amino acid. As I mentioned previously, there are only 20 standard amino acids involved in the makeup of all the thousands of proteins in our bodies. The unique properties of each protein are determined by the arrangement of these smaller chains, especially the R group, which acts as a variable. The bond between a carboxyl group and an amino group specifically is known as a peptide bond. A chain or combination of amino acids containing anywhere between 2 and 50 peptide bonds is known as a peptide. But when the chain of amino acids contains more than 50 peptide bonds, we call it a polypeptide. And finally, a complex molecule composed of one or more polypeptide chains gets folded into a three-dimensional structure to become a protein. And your DNA determines how the protein gets folded into its unique shape through a process we describe as gene expression. The shape of the protein is important because the shape dictates how the protein functions and how it interacts with other molecules in your body. For example, certain proteins are for structural support, while others are involved in immunity processes, and others still assist in biochemical reactions like enzymes do, or regulate bodily functions like hormones do, or transport other molecules like how hemoglobin transports oxygen in our blood. Proteins can also assist in muscle contraction, cell signaling, and other cellular processes. But it's the shape of the protein that ultimately determines what it can do. Unfortunately, our bodies can't produce proteins on our own. We must obtain them through our diet. However, it should be noted that the proteins we obtain from animals are already complete proteins, and the plant-based proteins are often partial or incomplete. So a vegetable-based diet requires us to combine different sources to get all of the essential amino acids and proteins that we need. For example, beans and rice are both plant-based foods that form a complete protein when eaten together. And there are other combinations that create complete proteins as well, but that's not the point of this video. So we've learned that cytokines are proteins that help move signals between cells. Cytokines are important in regulating immune responses by promoting or resolving inflammation. Now, inflammation is more than just expanding or swelling. It also involves redness, heat, and pain. Inflammation is usually present when your immune system sends white blood cells to an infected area to fight off an infection or a pathogen, and to initiate the healing response. And in this way, inflammation can be good, and it's typically a sign of healing or fighting an infection. However, inflammation can also be bad if it gets out of control. For instance, inflammation can cause fever or fatigue. It can lead to heart disease, cancer, and neurodegenerative diseases like Alzheimer's or Parkinson's disease. Arthritis and irritable bowel syndrome are inflammatory conditions that impact our joints and digestive tract. Anaphylactic shock is when your throat closes due to an allergic reaction that triggers inflammation in the throat. These are just a few examples of how dangerous it can be when inflammation gets out of control. Now that we understand what cytokines are and how they are involved in inflammation, let's talk about why scientists believe inflammation plays a role in developing autism. Inflammatory cytokines have been shown to lead to behavioral impairments reminiscent of autism during pregnancy. Persistent fever above a certain threshold in pregnancy has also been shown to increase the risk of autism in subsequent children. And fever, as we just learned, is directly tied to inflammation. Immune-related factors have also been associated with increased autism risk, and immune responses are also directly tied to inflammation. These reasons underscore the significance of the condition known as maternal immune activation, or MIA, which is a key autism risk factor. Now, MIA is when the mother's body triggers an immune response to fight off an infection. Now, this response inevitably leads to inflammation in the mother's body, but it has also been shown to directly impact the neurodevelopment of the fetus. Scientists at the University of Fukui in Japan compared the mother's blood to blood from the umbilical cord, and they have found that the plasma profiles are different in autistic children with regard to cytokines and metabolism. They even found that autistic boys had different profiles than autistic girls. And understanding these distinctions can help doctors identify the risk of developing autism very early without huge, costly tests and equipment. So, what are some of these differences that they observed in both samples? To answer that, we first need to understand a few more medical terms. Our bodies convert the food that we eat into small molecules known as metabolites, which are essentially the energy and the building blocks that our body uses for all of its functions. This process of conversion from food to metabolites is called metabolism. So, metabolism is our body's process for turning our food into energy and building blocks. And the better your body is at doing this, the better your metabolism. As we learned earlier, the building blocks for proteins are known as amino acids. And proteins are organic molecules involved in many cellular functions, including cell signaling. Fats, also known as lipids, are built using fatty acids. So fatty acids are also organic molecules like proteins, but they are involved in energy storage and cell membrane structure. Both amino acids and fatty acids are created by the process of metabolism. And I know fat is a bad word to some people, but try to remain calm. Not all fats are bad. Monounsaturated fatty acids, or MUFAs, and polyunsaturated fatty acids, or PUFAs, are both considered healthy fats. The difference between the two is simply that MUFAs have one double bond in their molecular chain, while PUFAs have multiple double bonds. A particularly important PUFA is arachidonic acid, or AA, which belongs to the family of omega-6 fatty acids. The chemical formula for AA is C20H32O2. Now, the chemical formula simply tells us how many of each kind of atom is in the molecule. So this formula means the molecule for arachidonic acid, or AA, contains 20 carbon atoms, 32 hydrogen atoms, and 2 oxygen atoms. And we can illustrate the molecule with a diagram like this. Now, this diagram includes labels for both of the oxygen atoms in red and the 9 hydrogen atoms that are double bonded. Carbon atoms are assumed to be in all the elbows. But where are the other hydrogen atoms? Well, carbon atoms have 4 bond points each, so the remaining hydrogen atoms are also assumed to attach to the remaining 23 carbon bond points, which you would see if we completed the diagram like this. And finally, the omega-6 part refers to the first double bond, which is located 6 carbon atoms from the omega end, or the end that's furthest from the carboxyl group. If you've followed along so far, this should all be making sense, but feel free to rewind if you are a bit lost. I'll wait. Okay, welcome back. AA in particular is produced in 4 positional isomers. Now, isomer is just a fancy word for a different arrangement of the molecule, but it doesn't affect the chemical formula. So basically, each carbon atom can still be arranged end to end in a long chain, but the hydrogen and oxygen atoms can be bonded at different places in the chain to make different isomers. This impacts the shape of the molecule, which, as we learned earlier, can impact how it functions in the body. This is the first isomer arrangement, known as the 5,6 dihet. This simply means the hydrogen and oxygen atoms are bonded to the 5th and 6th carbon atoms from the carboxyl group. When compared to autism test scores, these isomer levels did not have any significant correlation. Next is the 8,9 dehet isomer of AA, which has the hydrogen and oxygen atoms bonded to the 8th and 9th carbon atoms from the carboxyl group. Now, low levels of this isomer are correlated with repetitive behavior issues, especially in autistic girls. Then there's the 11,12 dehet, which has the hydrogen and oxygen atoms bonded to the 11th and 12th carbon atoms from the carboxyl group. High levels of this isomer are correlated with social behavior issues, especially in autistic girls. And the fourth isomer is the 14,15 dehet, which has the hydrogen and oxygen atoms bonded to the 14th and 15th carbon atoms from the carboxyl group. This isomer is involved in lipid or fat metabolism and inflammatory signaling. Higher levels of this isomer strongly correlated to higher autism test scores in both boys and girls. In simple terms, lower levels of 8,9 dihets correlated to higher levels of 11,12 dihets, and also the higher the levels of 11,12 dihets or 14,15 dihets, or the total dihets altogether, correlated to higher autism scores, especially in autistic girls. Studies have shown that autistic children have elevated levels of polyunsaturated fatty acids, or PUFAs, in their systems compared to their neurotypical counterparts. And these elevated levels were especially apparent in three major enzyme pathways, which we'll get into shortly. At this point, it's important to note that a higher ratio of fatty acids puts us at risk of inflammation, which means we are at higher risk of developing conditions such as arthritis, allergies, asthma, cancer, cardiovascular disease, inflammatory disease, autoimmune disease, and more. So this is a significant finding, and it can explain why so many autistics have co-occurring health conditions. If scientists can get the inflammation under control, it could help autistics lead healthier lives by lowering our risks of these conditions. Very cool, right? Now, I just mentioned enzyme pathways. And in case you don't know, enzymes are proteins that speed up chemical processes and chemical reactions in the body. Without enzymes, biological processes like digestion, cell growth, and muscle function would either occur more slowly or not at all. Now let's talk about the three enzyme pathways that stood out to the Fukui researchers. Here we have our arachidonic acid represented by a car. And the path that the car takes determines what the AA fatty acid will end up being metabolized into. The forked road heading to the left is for the enzyme known as cyclooxygenase, or COX. The forked road heading to the right is for the enzyme known as cytochrome P450, or CYP. And the forked road heading straight ahead is for the enzyme known as lipoxygenase, or LOX. Let's talk about the destinations that these forked roads or enzyme pathways lead to. Cyclooxygenase, or COX, is an enzyme pathway that can convert AA into prostaglandins, which are hormone-like substances usually produced at the site of an injury or infection. And these substances are involved in inflammation, pain, fever, blood clotting, and other various physiological processes. Medicines like aspirin and ibuprofen work by inhibiting prostaglandin synthesis to reduce inflammation and pain. COX can also convert AA into thromboxanes, which are crucial to the formation of blood clots by causing blood vessels to constrict and encouraging platelets to clump together to stop bleeding at the site of an injury. And aspirin reduces thromboxane production to prevent excessive blood clotting, commonly associated with heart disease. Lipoxygenase, or LOX, is an enzyme pathway that converts AA into hydroperoxyeicosatetranoic acids, or HPEATs, which are further metabolized into eicosanoids. Eicosanoids are signaling molecules involved in various biological processes, such as inflammation and cell growth. Eicosanoids related to LOX include leukotrienes, or LTs, which are potent inflammatory mediators, particularly involved in asthma and other inflammatory diseases. Lipoxins, or LXs, can have anti-inflammatory and pro-resolving effects. Hydroxyeicosatetranoic acids, or HEATs, essentially the H-peat without the peroxide portion, it can have diverse roles in inflammation and cell signaling. Now, cytochrome P450, or CYP, is the third enzyme pathway, and it can convert AA into epoxy fatty acids, such as epoxyeicosatetranoic acid, or EET, or EAT. Now, EATs have anti-inflammatory effects and are involved in various processes, including blood pressure regulation, inflammation, and cell signaling. CYP can also convert AA into hydroxyeicosatetranoic acids, or HEATs, which are eicosanoids involved in inflammation and cell signaling. Yes, these are the same as the HEATs created through LOX, but they can also be formed through CYP. Okay, stick with me just a little longer. We've finally gotten through the learning portion of this video. Now let's talk about what came from this study. Researchers in Fukui found that converting polyunsaturated fatty acids such as AA using the CYP pathway may impact fetal development during pregnancy in the presence of maternal immune activation, or MIA. Specifically, the CYP pathway that leads to the metabolization of EATs has been shown to contribute to neurite growth, which means it helps neurons develop new projections and extensions, allowing for more connections. So although EATs are typically anti-inflammatory, they can result in more connections and branches in the brain, and the inflammation triggered during MIA significantly impacts neurodevelopment by counteracting the anti-inflammatory effects of EATs. But that's not all. We know genes account for 80 to 90% of the risk for developing autism, and there are several genes implicated in the development of autism. Specifically, EPHX2 genes have been observed to be more prevalent than average in the postmortem brains of autistic patients. This is important because EPHX2 genes encode soluble epoxide hydrolase, or SEH, which is another important enzyme that takes the epoxy fatty acids, or EET, and further metabolizes it into dihydroxy fatty acids, or diols. One such diol is known as dihydroxyeicosatetranoic acid, or DHET, or DIHET-REs. While EATs are generally anti-inflammatory, these particular diols that it converts into have inflammatory properties that regulate vascular tone, blood pressure, renal transport, and other cellular functions. Now, the isomers of diheats affect the fetus the same way isomers for AA do. So, for example, lower levels of 8-9 diheat levels impact repetitive behaviors, and higher 11-12 diheat levels impact social behaviors. This means that researchers have discovered autism develops in one or more of the following ways: lower diheat R levels, and or raised diol levels, and or increased SEH activity. And these processes occur during the fetal period, but affect autism symptoms or difficulties with daily functioning after birth as well. These findings suggest that the presence of CYP-PUFA metabolites during the fetal period plays an important role in generating a biological background that leads to the development of autism-like behavior, especially in autistic girls. Now, these differences were very apparent in the umbilical blood samples that were taken immediately after delivery. However, the differences between boys and girls nearly disappeared in the autism tests taken 6 years later. And researchers suspect that this may be due to the fact that girls tend to be better at adapting their communication and social behavior, and that suggests that the effects of diheats involved in MIA may be important to the development trajectory for autistic children after birth. To recap, we have the mother eating polyunsaturated fatty acids, which are converted into EATs with the help of the CYP enzyme pathway. This results in development of more extensions and projections in neurons, allowing for more brain cell growth and brain cell connections. Then she has an immune response, which increases inflammation. Then the gene EPHX2, which is more prevalent in autistics, creates soluble epoxide hydrolase, or SEH, which further metabolizes the EATs into DHETs, or dihet-REs. And that leads to further inflammation. And this is particularly significant for autistic girls. It's like the perfect storm of inflammation. It confirms earlier hypotheses that inflammation plays an important role in the development of autism. That's why this discovery is such a huge deal. But we're not done. There's more. And other known issues impacting inflammation can still play a role here. For example, glyphosate is a widely used herbicide that controls broadleaf weeds and grasses. It has been registered as a pesticide in the US since 1974. The Fukui study found that exposure to high concentrations of glyphosate during pregnancy and lactation induces autism-like behavior, along with significant decreases in 8,9-diheat levels, which worsen repetitive behaviors, and significant increases in SEH levels in the plasma and multiple brain regions, which we now know leads to more production of diols that promote inflammation. To test their hypothesis, researchers took interleukin-10, or IL-10, which is a cytokine that's shown promise in its ability to inhibit the production of pro-inflammatory cytokines, and they injected it into pregnant mice who were exposed to glyphosate. And they found that this improved autism-like behavior in their offspring, such as increased grooming time and improved social interaction. MCP1 is another cytokine that affects the movement of white blood cells towards sites of inflammation. Low levels of MCP1 are associated with worsened sociality and anxiety. Now, similar to the process we discussed for AA, MCP1 is converted into EATs through CYP, and then further metabolized by SEH to form diheats. The cytokine IL-6 is also associated with autism-like behavioral abnormalities that develop during maternal immune activation, or MIA. And IL-6 is dependent upon MCP1. Therefore, researchers conclude that diheats may influence the development of autistic symptoms and adaptive functions via inflammatory cytokines like MCP1 and IL-6. Now, I know the possibility of eugenics is always a concern when we hear announcements such as scientists discover the cause of autism. However, the results of this study are not focused on eliminating autism. They are focused on preventing what they call severe autism. And there's a big debate in the autism community over whether severe autism even exists. Many autistics believe you are either autistic or you aren't. And the support levels are really about your individual support needs. But that's precisely what researchers in this study are talking about. Those who struggle more with communication, repetitive behaviors, and inflexibility have been shown to test higher in autism diagnostic tests, and those scores correlate to findings related to the dye heat levels and the EAT levels that were found in autistics, particularly autistic girls. So their findings may finally be settling the debate about the existence of severe autism or not. So what is next? It's hard to say where research will continue, but let's try to make some informed guesses. Considering EATs have anti-inflammatory properties, scientists are not likely to focus on suppressing synthesis of EATs when their concern is inflammation. This is good for autistics because EATs contribute to the additional branching and connections in the brain, which is typical of autism. Therefore, it's likely we will continue to see autistic people being born for the foreseeable future. However, scientists are likely to focus on suppressing SEH from synthesizing inflammatory diols. In fact, they've already found an enzyme known as ARG287GLN, which shows some promise with its ability to reduce SEH activity in cells. Unfortunately, dye heats do not pass through the blood-brain barrier, so researchers are unlikely to focus on dye heats directly, but would rather focus on precursors to dye heats, like SEH. And that wouldn't necessarily eliminate autism so much as it would eliminate higher severity levels and co-occurring conditions, particularly in autistic girls. To answer the initial question as to whether scientists actually discovered the cause for autism or not, the answer is no. That was simply clickbait for the articles. This study focused on inflammation and metabolism, and it's highly unlikely that these mechanisms account for the full 10 to 20% of environmental factors associated with autism development. So while this gets us closer to understanding autistic development on a cellular level, there's still so much more we need to understand and confirm before we can definitively say that we have identified any cause of autism, let alone before claiming that there's a singular cause, if there even is one. Now, even this study does not claim to have found a singular cause, considering there was very little correlation between blood samples and autism tests results for autistic boys. So don't worry, there's still a lot more research that needs to be done. Meanwhile, the discovery at the University of Fukui seems to be promising in that it should make earlier diagnosis possible at a more affordable price tag. And the results of those tests would likely be more consistent and come with a higher level of confidence than today's diagnostic procedures, which can be highly subjective. And that opens the doors for better identification of autism, better supports, and earlier interventions. And all of that is good news. So I hope you learned something new from this video, and I especially hope that you got more from this than those generic social media posts. As more advancements are made in how autism develops, I'll be sure to follow this up with another video. But until then, thank you for watching. If you enjoyed this video, please comment, like, and subscribe to see more content like this, or click on the video here to continue watching.